and welcome back to day 3 of Live from the Lab. I'm Rachel and I'm going to show you around a bit more, and we're hopefully going to find some more scientists to talk to. I just wanted to answer one of the questions. It's been great that you've been sending them in. So we had a question about how we became scientists and do we need to be clever? And I think being clever is useful, but also being creative and wanting to learn more is great. So make sure you keep doing all those fun experiments at home and keep going to school and enjoying all the science lessons. So I'm going to have a little wander now and see who we can see, and you can come with me. Oh, hi Dr. Harry, how are you today?
Oh, hello Rachel, I'm very good, thank you.
What are we doing?
So I need to process some samples here. I have some serum samples. So these are from blood, from human blood, and I need to freeze them.
Doesn't freezing take hours and hours?
So sometimes it does, but here in the lab we can speed up this process. So here today I'm going to use some dry ice to speed up the process. So how long do you think it's going to take to do it using the dry ice?
Wow. Overnight?
No, actually it's going to take only 2 minutes. Do you want to see it?
Yes, please. That's really magical as well, like Harry Potter.
Yeah, isn't it? So first of all, we need to be very careful with the dry ice because it's really cold. So the temperature here is about minus 78 degrees. So we need to be very careful on not burning our hands. So here we go. So as you can see, we have our samples in the liquid form, and I'll put in the dry ice right now. Very good. So they're all coming here, and now we can start the timer, and it should be done in about 2 minutes.
So how come we need to freeze our samples in the lab?
So it's needed because I want to keep my samples safe to be analyzed in the future. So in the future I'll be able to quantify several molecules that it's in the blood, but I cannot do it now because I want to process several samples all together. So I need to keep them safe for as long as it's possible, so that in the future myself or other researchers can actually quantify things like hormones, like other proteins, in these samples here. And it really needs to be frozen. The process of freezing needs to be quite fast because I want to avoid any degradation. I don't want my samples going bad. Do you want to see where I'm going to keep my samples afterwards? Because I cannot keep in the dry ice, but I can transfer to a proper, very good freezer that we have in the lab.
Yes, please. We actually had a question from George asking what was stored in all of these big fridges and freezers we have around.
That's fantastic. So I can show you now. So here I have a minus 20 degrees Celsius freezer. So this freezer here is exactly the same as the one you have at home. So where you usually keep your food. So here we have everything frozen at minus 20, and as you can see, we're gonna have like several reagents that we might need to use in the lab for experiments. We have samples. It could be biological samples, chemical samples that also needs to be protected from going bad. So that's why we need to keep at low temperatures. But there are some samples that are very precious, and we don't want to take any chances that it go bad. So what we need to do is to keep at even lower temperatures. And then for that, we can use a different type of freezer, which is a minus 80 degrees Celsius freezer. So that's very cold. So you can have, for you, like to have an idea, this is colder than winter in Antarctica. It's
wow.
Very cold.
That's cold.
Do you want to see it?
Blood. Yes, please.
So again, in this type of freezer, we're gonna put different types of samples, biological samples. We're gonna put some reagents that we need, and then we're gonna stay here very cold for a very long time, so we can keep everything well protected until we need to use in the future.
Cool. Very cool.
Shall we go back to our samples now?
Yeah, let's see if they're frozen. How long do you think you can store samples for?
So it depends on the sample. But years and years most of the time. If we're using a freezer like the -80, we can definitely keep it there for years. Can you see it? So absolutely frozen. So now it's solid again.
Wow, ready to be stored and analysed at a later date.
Absolutely. So now the only thing we need to do is just transfer it to the -80.
That's great. So I hope that answers your question, George. So all of these fridges and freezers in the lab are full of people's samples, ready to be analysed when we have the time and the methods ready.
Here we go. Lovely. Safely stored.
Great.
Thanks, Rachel, for coming by. Well, what are you going to do next?
So I'm going to explore a bit more. I might go and find Orla. I think she's doing some cool stuff on the microscope.
That's good to know. Can I ask you a favor?
Sure.
I have some sample that I really need to give it to her. Would you take it?
Yeah, of course.
That's fantastic. So I have you in my fridge, and I asked her a favor that she could look in her microscope some samples for me.
Of course, yeah, I'll take these down.
Thank you very much.
Thank you. Bye, Dr. Harry.
See you soon. Bye, guys.
Come with me. Let's go and find Orla at the microscope. Hi, Orla. How are you?
Hi, I'm good, thank you. How are you, Rachel?
Yeah, good. I've got some samples here from Dr. Harry. He told me to bring them down.
Oh, that's perfect. Thank you so much.
So what are we working on?
So, in my research, I look at how the brain controls what, when, and how much we eat. But of course, the human brain is far too complicated to study in the lab. So I use mouse brains. So if we have a look at these slides, here we have some mouse brains. Can you see those? So in these mouse brains, what I've done is I've injected a virus, but not the type of virus that causes COVID-19. This is a modified virus, which is only survives and replicates in specific cells within the mouse brain. And what I want to do with this virus is look at how these particular cells are connected with other regions within the brain. So, should we have a look at this slide and see if we can see any of my virus in the cells?
Yeah, that would be great.
Okay, let me just load it onto our confocal microscope. So with this microscope, this uses lasers to illuminate our cells because they're very small, so we can only see them under a specific light. Okay. There we go, all lined up. Right, so if we click live here on our program, and then we click these different channels,
Look at all those colours.
we can see our different colours.
That's great. We had a question about what happens if we mix blue and green together. So can you explain what the different colours mean?
Yes, of course. So here in blue, we've got a blue dye which shows where our different brain cells are. In green, this is a green fluorescent protein, which is normally found in a jellyfish, but here it's found in specific brain cells. And then in red, we show where the virus is in our brain. And then in the final picture here, we have an overlap between our red virus positive cells and our green brain cells. So there you go.
So thanks for joining us. This has been day 3 of Live from the Lab here at the IMS. Make sure you come back to watch day 4 tomorrow and send us in lots of questions and check out our website for any other fun Science Festival events. Thank you.